Hi everyone, and welcome to the lecture video for the first day of class of BME2104. Today we're going to talk about a parts list for the cell and the central dogma. These are fundamental concepts that we're going to use in all the other lectures for the rest of this course, so I want to make sure that we lay down a really solid foundation with this lecture. As we go through the lecture, make sure to think about questions you can ask during class, and please fill out the survey to indicate which slides in this presentation you have the most questions about. We're going to start today talking about cell diversity and cell theory, followed by going over the basic molecular building blocks that form a parts list for the cell. I'll also cover the major structures inside the cell, including many different types of organelles, and then we'll move on to the central dogma. For every lecture in this course, I'll use an example from human disease to illustrate key topics and concepts in the course material, and this semester, of course, we're going to focus very heavily on COVID-19. Today, we're going to start out our focus on coronavirus by asking the very basic question: what is life, and how do viruses like the coronavirus fit into this definition? So let's start now with an introduction to coronavirus and the pandemic that we're all living through. The disease called COVID-19 is a contagious respiratory illness caused by the coronavirus SARS-CoV-2. That's short for Severe Acute Respiratory Syndrome Coronavirus 2. It's named 2 because we had a similar coronavirus outbreak that caused severe respiratory illness back in 2003, although that one was better contained and didn't turn into a worldwide pandemic. Coming back to the present, SARS-CoV-2 was first observed as an outbreak in Wuhan, China back in December 2019, but since then it has spread all over the world as a truly global pandemic. On this slide, we're going to trace the spread of coronavirus around the world. On the left, this plot shows the number of new cases per day, and the map on the right shows where the cases are most concentrated over time, going from February right up until the middle of August. Two obvious things to point out are this big spike in late March centered around Italy and Spain, and then the continued growth and spread to many other countries, dominated in large part by our own country, the United States. Zooming in on the US, we can see the same initial spike in late March, which was largely centered in New York City, New Orleans, and Atlanta, followed by more diffuse spread throughout the rest of the country, with a peak in new cases coming around the 4th of July holiday. And this is our current status on August 16th. These plots are up to date now while I'm recording this video, but if you're watching it later and want to see a more updated version, I've included links that you can follow on this page. Also, at the end of the class, we can all come back and see how we're doing. Hopefully things will look better than they do now. The plot in the bottom right is actually something I made myself. I saw a similar animated plot online, and I really liked the red spike visualization, but it was from June, and they never updated it with new data, so I decided I would just make my own version of this plot with publicly available data. One thing to note for this plot is that it's showing the infection rate per county and the number of people per 1,000 who were infected, rather than the total number of infected people. When I have time, I'll put my code for this up on GitHub and add that link to these lecture notes so you can track it yourself if you're interested. Also, if anyone's interested in doing a little extra coding themselves, it would just take a few changes in my code to make the same kind of plot for the state of Virginia, maybe including surrounding areas like DC and North Carolina as well. Alright, so that's how the coronavirus pandemic is spreading. Let's talk now about what having this disease actually feels like. Symptoms of COVID-19 include fever and mild to severe respiratory illness, with symptoms such as coughing and shortness of breath. We've also seen headache, sore throat, fatigue, and diarrhea. One mysterious symptom that has been noticed more recently is the loss of taste or smell. Another important thing to note is that many individuals can carry and spread the SARS-CoV-2 virus without displaying any of these symptoms. This is referred to as asymptomatic spreading and has made the pandemic particularly hard to control. Many people who are infected with SARS-CoV-2 will recover after experiencing relatively mild symptoms, but a significant fraction of infected individuals will experience more extreme symptoms requiring hospitalization and in many cases death. Looking at official statistics, we've detected 5.4 million SARS-CoV-2 infections in the United States and recorded 170,000 deaths, putting the fatality rate in our country at approximately 3%. In these cases, viral infection in the lungs leads to cell lysis and immune response, and the resulting inflammation and tissue damage leads to pneumonia, which is characterized by the alveoli in the lungs becoming filled with liquid. This makes it difficult to breathe and less oxygen enters the bloodstream. This lack of oxygen, or hypoxia, causes massive damage to tissues throughout our body, leading to organ failure in the liver, kidneys, and other vital organs. And this is the main cause of fatality in COVID-19. Okay, that's our introduction for COVID-19. We'll talk a lot more about different aspects of this disease over the course of the semester and track our progress in fighting the pandemic as well. At the end of today's lecture, we'll go over some basic definitions for viruses, walk through the SARS-CoV-2 viral life cycle, and talk at the end about how viruses fit into our definitions and ideas about life. Let's move on now to the main lecture material and start out by talking about cells. We're going to start out talking about cells because they're the basic unit of life, just like atoms are the basic unit of chemistry. What do I mean by this? Every living organism can be broken down into its individual cells, and as we'll see in this class, thinking about organisms at the cellular level is usually the best way to understand their physiology and disease processes. There's a large number of single-celled organisms, like bacteria, for example, but in this class, we're going to focus mostly on multicellular organisms, such as ourselves. The human body is made up of around 40 trillion different cells, and it's this great diversity of cell types in our body that gives us the ability to do everything that's important for our survival, such as eat, breathe, adapt to our environment, and reproduce, just to name a few. Just to give you an idea about the great diversity of cell types we have in our body, I'm showing here a scale illustration of a neuron cell, which has this tree-like branching morphology and can extend up to 1 meter long in our body. And next to that, we have a lymphocyte cell, which is small and rounded, with a diameter of approximately 10 microns. Above that, I'm showing a picture of a typical mammalian cell, shown by cross-section to reveal all the different internal structures called organelles. We'll go over all of these different organelles later in the lecture. But first, let's talk very briefly about cell theory. This is a very old theory, starting out with the development of the microscope, which allowed our first observation of cells in the 1600s. In the 1800s, the centrality of cells to life became more apparent, and the following cell theory was developed. First, that all living things are made up of cells. Second, that cells are the basic unit of life, similar to how atoms are the basic unit of chemistry. And third, this is the one that took longer to nail down. The third one is that all cells arise from other cells. This is an old theory, but it still holds up very well today, and it's a valuable reminder that as biomedical engineers, when we think about a biological system, the best way to understand it is usually going to be to focus on the cells. As I mentioned earlier, there's a huge diversity of cell types between different organisms and even within the same organism. For example, this diagram on the left illustrates the wide variety of cell types that can be found within the human body. All of these cells are made up of the same molecular building blocks, but the way these building blocks are put together can be very different, giving rise to all the different cell types. It's kind of like Legos. You can have a very basic set of Lego bricks, but then build pretty much any structure you want with them. The building blocks for a cell consist of 4 major classes of biomolecules: lipids, proteins, carbohydrates, and nucleic acids. We'll go over the structure and properties for each of these biomolecules over the next few slides, but first, I want to give you a picture of how much these contribute to the overall cell mass. This figure is for a bacterial cell, but it's not so different from eukaryotes like us. As you can see, the majority of any cell is made up of water, 70% here. The remaining 30% of material are broken down as follows: 4% for ions and small molecules, which we're not going to really focus on today, 2% for phospholipids, the major class of lipids in our cell membranes, followed by 1% DNA and 6% RNA, so 7% total for nucleic acids. Proteins are at 15%. You can also think of this as 50% of the dry mass of the cell. And finally, we have 2% polysaccharides, the major form of carbohydrates in our cells. So that gives you the average makeup of a cell. I want to quickly point out that this figure comes from a textbook called Molecular Biology of the Cell, which is available free online through NCBI Bookshelf. I don't use a textbook for this class, but if I did, it would definitely be this one. I put the link right here so you can check this out if you're interested. All right, so we're going to go over all the molecular building blocks of the cell, but before we do that, I need to go over how we're going to draw them. We use a visual shorthand called skeleton structures, which many of you have probably seen before in general chemistry or organic chemistry. In the skeleton structure, carbon atoms are not drawn, and hydrogen atoms that come off of carbon atoms are also not drawn, leaving us with stick figures like this in their place. We do this because there's just too many carbon and hydrogen atoms in all the biomolecules we're going to cover in this course. If we drew them all out, it would take up too much room on the page, and it would take too long if I was drawing these on a dry erase board. So we use this skeleton structure visual shorthand instead. Double lines are used to indicate double bonds, as shown in this benzene molecule that we'll see on Thursday in the amino acid phenylalanine. And all the other atoms besides carbon and hydrogen are drawn with their 1 or 2 letter abbreviation, as shown in this pyrimidine molecule that we'll see later today in nucleotide bases. As you'll see in the upcoming slides, these strict rules for drawing skeleton structures aren't always followed precisely. Sometimes the hydrogens will be left in to emphasize bonding interactions they participate in, or to emphasize the presence of single versus double bonds. But it's important for you to understand this general convention so we can all be on the same page when we're discussing biomolecular structures. So the first molecular building block for cells that we're going to discuss are called lipids. There are a few different categories of lipids that are important for cell biology, and we'll go over these in more detail in our lecture on lipids next week. But for now, we'll start out by considering 2 major lipid categories, the first being fatty acids. These are long linear chains of carbon atoms with a COO carboxylic acid group at one end. The carboxylic acid group is polar and somewhat reactive chemically, but the major property of fatty acids is their hydrophobic carbon tails. Hydrophobic meaning they don't interact favorably with water. Remember that cells are 70% water, and in this largely aqueous environment, fatty acids repel water and form fat droplets. There's some cells in our body that specialize in fat storage, and these will have very large fat droplets that take up most of the cell volume. These are an extreme exception to the general rule that most cells in our body are around 70% water. All right, so that's fatty acids. Another really important category of lipids that form a building block for our cells is called phospholipids. As you can see in this illustration on the bottom, these are actually composed of 2 fatty acids linked together plus a phosphate containing polar head group. Phospholipids are really important because they make up the membranes in our cell, like the plasma membrane, which we'll talk about later in this lecture. The properties of these lipids are driven by hydrophobic interactions and the geometry of their structure. We'll go over this in more detail in our lecture on lipids next week, but briefly for now, fatty acids in aqueous solution form spherical aggregates called micelles to pack all of their hydrophobic tails together. The polar carboxylic acid groups form a surface of the micelle to interact with the surrounding water molecules. The same hydrophobic driving forces control the properties of phospholipids, but because their geometry is a little different, they tend to form lipid bilayers, where hydrophobic tails are buried in the middle, and the phosphate-containing polar head groups form a surface that interacts with the surrounding water molecules on each side. One last thing to talk about lipid bilayers is that they can exist as sheets in certain experimental setups, but generally a phospholipid bilayer sheet will be, if it's just as pictured on the top in this illustration, that would be very unstable because all the hydrophobic tails are exposed along its edge. So what we see in cells is lipid bilayers folding into spherical surfaces like this, which is much more energetically favorable because all the hydrophobic tails are now buried and protected. All right, so that was lipids. Now let's move on to proteins. We'll cover this in much more detail on Thursday, but for now, let's just go over a few basics. Proteins are linear polymers of amino acids. A generic amino acid is shown here. All amino acids have an amine group, shown in blue, and a carboxylic acid group, shown in red, and these are linked together by a single carbon atom in the middle. What makes amino acids different from each other is their unique side chain, indicated here by the generic R. This is a shorthand used in organic chemistry, kind of like X is used as a variable in mathematics. In pretty much every organism that we'll talk about in this class, there are 20 standard amino acids that look just like this, each with a different R group with different chemical properties. The thing that makes amino acids a really great building block is that they can be linked together by a condensation reaction between the amine and carboxylic acid groups. This new linkage is called a peptide bond, and it can be done over and over again to create long linear polymers of amino acids. When these are short, we call them peptides, and when they're longer, we call them proteins. There's not a hard and fast rule about this, but I'd say somewhere between 30 and 50 amino acids is around the dividing line. In aqueous environments, such as inside the cell, These long linear chains tend to fold up into three-dimensional conformations, and this structure will play a big role in determining the function of that protein. Like I said earlier, there are 20 standard amino acids which have R groups with very different chemical properties. And using these building blocks in different combinations, this can give us a huge variety of proteins that serve different functions in the cell, including enzymes, structural proteins, cell signaling proteins, and transporters, just to name a few. In my opinion, these are the real MVPs of the cell, and we'll talk about them a lot more in our next lecture on Thursday. All right, the next molecular building block to talk about is carbohydrates. These molecules are used for energy storage, and it's easy to remember their chemical formula because it's just carbon and then a water molecule. That's why they're called carbohydrates. The most stable carbohydrates are 3-carbon, 4-carbon, and 5-carbon molecules. This diagram shows 3 carbohydrates that we'll see again later in the course. 4 and 5-carbon carbohydrates can be further stabilized by cyclization. I'm showing here the interconversion between the linear and cyclic forms of the 6-carbon carbohydrate called glucose, as well as some other representations of glucose with the ball and stick, space-filling model, and a boat diagram in the bottom right that some of you may recognize from organic chemistry. I'm showing a lot of stuff here. Don't worry about memorizing these structures or names of different carbohydrates today, but I do want you to know that these different carbohydrates can be linked together into short or long polymer chains. Another word for these carbohydrates is saccharides. They're really just different kinds of sugars, and these saccharides can exist as monomers called monosaccharides, dimers called disaccharides, or longer polymers called oligosaccharides. Usually these will be with 6-carbon sugars in the cell. Here's an illustration of the polymerization reaction between 2 saccharides, glucose and fructose. Just like with amino acids, this is a condensation reaction, which just means the loss of a water molecule. One thing that's very different from amino acid polymers, though, is that oligosaccharides are not limited to linear chains, but can instead have very complex branched structures. This is because they have multiple hydroxyl groups that can participate in polymer bonding. In the picture of glucose and fructose, in the picture of the glucose-fructose disaccharide, you can see that each monomer still has 4 other hydroxyls that could potentially be used for additional linkages. One place where we see a ton of branched oligosaccharides is on the outside surface of a cell. This effectively forms a soft, sugary coat around the cell, and this comes from oligosaccharides covalently linked to membrane lipids and membrane-localized proteins. The last molecular building block for the cell that we're going to talk about today is nucleic acids. These include DNA, which is used for genetic information storage, and RNA, which is used for protein translation, a form of genetic information retrieval. Both DNA and RNA are long linear polymers of nucleotides. A nucleotide is this slightly complicated molecule, so let's go through the pieces of it one at a time. Every nucleotide has a sugar molecule, aka carbohydrate, at its core. This is shown in blue in the diagram. RNA has a sugar ribose. The name RNA is short for ribonucleic acid. DNA is short for deoxyribonucleic acid. It'll look just the same as this, but its sugar, deoxyribose, is just missing the OH hydroxyl group from the lower right side. All right, so that covers the sugar part of the nucleotide, but there are these other important components as well. Coming off the 5' ribose hydroxyl on the left is a phosphate group. You can see here that it has 2 negative charges. This is why we call them nucleic acids, because the phosphate group is acidic. In this picture, it's already given up 2 of its protons into solution. On the other side of the sugar core, we have a base group coming off the 1 carbon. So every nucleotide will have these 3 components: a phosphate group, a sugar, and a base. DNA and RNA form long linear polymers by forming covalent linkages between the 5' phosphate and the 3' hydroxyl. Just like the peptide bonds and oligosaccharide bonds, this bond is also formed by a condensation reaction. In every cell in our body, we have long linear DNA polymers that are over 200 million nucleotides long. Information is stored in the linear order of bases. DNA has 4 different bases: adenine, cytosine, guanine, and thymine. RNA also has 4 different bases. The first 3 are the same, but the last one is uracil instead of thymine. The structure of nucleic acids plays an important role in how it allows for information storage and retrieval. DNA is most stable in aqueous solution as an antiparallel double helix that's held together by hydrogen bonding between complementary bases. We refer to these as base pairs. Adenine fits with thymine, forming 2 complementary hydrogen bonds, and guanine fits with cytosine, forming 3 hydrogen bonds. This figure shows a 3-dimensional representation of the DNA antiparallel double helix, in space-filling form on the left and drawn with our skeleton line convention on the right. You can see that the base pairing happens in the middle of the helix, and the bases are flat and stacked on top of each other, kind of like stairs in a spiral staircase. We'll talk more about the antiparallel nature of the double helix later in the semester when we get to our lecture on DNA replication. The genetic information stored in DNA is like the architectural plans for the cell, and what we're going to talk about now is how those plans are read out and implemented. This is the central dogma, which describes the flow of information in the cell, from DNA to RNA to protein. The order of nucleotides in DNA is read out and converted to RNA by a process called transcription. This is an apt name because what we're basically doing here is rewriting the same nucleotide sequence, but with uracils instead of thymines. The next step, going from RNA to protein, is called translation, and this name is also apt because it really is like going from one language to another, from nucleotides to amino acids. This was one of the big challenges for biology in the middle of the 1900s, figuring out how the sequence of nucleotides in DNA and RNA could be translated into the amino acid sequence in proteins. It turns out the answer to this puzzle was that every 3 nucleotides codes for a single amino acid. These nucleotide triplets are called codons, and because there are 4 different nucleotides in DNA and RNA, that means that there are 4 to the third, or 64 different possible codons. This table shows every nucleotide triplet codon and which amino acid it codes for, given by the amino acid's 3-letter abbreviations. We'll come back to these on Thursday. I definitely don't want you to memorize this table, even though it's blue, but there are 2 things that I want you to take away from this. First is that the genetic code is degenerate, which means that almost all amino acids are coded for by more than 1 codon. When you think about it, it has to be this way because there are 64 codons but only 20 amino acids. Second, I want you to take away from this that there are specialized codons that don't code for any amino acid, but instead code for the start or stop of protein synthesis. The start codon AUG actually does code for an amino acid, methionine, and this makes for an interesting quirk of evolution that every newly synthesized protein will start its amino acid sequence always with a methionine. Like I said, I don't want you to memorize this table, but we will come back to all 20 amino acids and their different properties in our next lecture on Thursday. Okay, just to repeat for emphasis because it's so important, the central dogma describes the flow of genetic information in cells from DNA to RNA to protein. One more point to quickly add is that in addition to being transcribed, DNA can also be replicated. This is what happens every time a cell divides, and we'll talk about that later in the semester as well. One more important thing to mention is that RNA molecules that encode proteins have a specific name, and this is messenger RNAs. Inside of our cells, DNA exists as long deoxyribonucleotide polymers. These long DNA molecules and their associated proteins are called chromosomes. Humans have 23 linear chromosomes, ranging in length from 50 million to 250 million nucleotides each. If we took all 23 chromosomes from a single human cell and stretched them out in their linear double helix form, their length would be 1.8 meters, or around 5 feet. It's pretty amazing to think about a molecule that long being packed inside a single cell, or even more impressively, being packed inside a compartment within the cell called the nucleus, which is only 5 microns in diameter. The way this tight packing occurs is illustrated on the right. You can see there are multiple levels of organization to achieve this amazing level of compression. On each chromosome, there are approximately 1,000 genes, and we'll talk more about what a gene is on the next slide. When we take all of our chromosomes together, the entirety of our genetic information, this is referred to as the genome. So let's talk a little more now about genes and their structure. This is another important concept, and I really want to make sure that we lay a strong foundation for the rest of the semester. Genes are linear stretches of DNA on our chromosomes that encode proteins or other gene products. Generally speaking, each gene corresponds to a specific stretch on the chromosome, but there are many cases where genes overlap with each other, and a few examples of this are shown on the right. A key thing to remember here is that a gene will occur on one strand of the DNA helix or the other, so sometimes you can have overlapping genes on the antiparallel strands, as shown in B and C of this figure. One more thing I want to talk about is the structural organization of genes. These will typically have regulatory DNA sequences at the ends, and then protein coding DNA sequences in the middle. Okay, so now we've covered the molecular parts list for the cell and the central dogma. In the next set of slides, I'm going to go over the major structures of the cell, listed here on the right. I won't spell them all out now because we're going to go through them one at a time, but I do want to quickly draw your attention to the illustration on the left. It's by one of my favorite authors and web cartoonists, Randall Munroe, and what it's doing here is explaining all the structural components we're about to talk about using only the 1,000 most common words in the English language, just as a fun exercise. So for example, it seems that cell isn't one of the top 1,000 words in the English language, so instead they're referring to cells as tiny bags of water that you're made of. We won't be using these simple definitions in class, but it's fun to see how you can use simple words in creative ways to describe all the components we're about to talk about. All right, so here we have a nice overview diagram that contains all the structures we're going to cover today. Before we go over these individually, I want to talk about the general term organelle. Remember that in lectures for this class, any text colored in blue is used to indicate things that you should focus on while you're studying. So let's give a definition for organelle. This is a membrane-enclosed compartment within a eukaryotic cell that has a distinct structure, macromolecular composition, and function. Not everything labeled in this diagram is an organelle, but here are the organelles we're going to focus on today. We'll also cover these other important structures, including ribosomes, the plasma membrane, and different parts of the nucleus. One thing that's important to keep in mind when we're looking at pictures like this is that it's just an artistic rendering of a cell. In biology research, we often refer to figures like this as a cartoon. This cartoon happens to be pretty good in terms of its spatial layout and dimensions, but it's always better to look at the real thing. So whenever possible, I'll show you experimentally determined images and structures for the things that we're talking about in this class. This figure on the right is a transmission electron microscopy image of a mammalian cell, and in this image, you can see most of the major organelles we're going to discuss. One more thing that we'll do for each of the organelles and other structures that we discuss is go over their size. So to put those numbers in perspective, let's start out with the size of an entire cell. These can vary quite a bit, as we've already seen with the example of neurons and lymphocytes, but a quote-unquote typical mammalian cell will be on the order of approximately 10 microns in diameter. We'll go over all these numbers again on a summary slide, but I want to call your attention here to the fact that I've highlighted this number in blue. All right, so now we're going to start going through the most important structures in the cell one by one. For each of these structures, I'll highlight its location within the cell on the bottom left in magenta, and I'll present cartoon and experimentally determined images on the right. We're starting out here with the plasma membrane. If you ask me, this is probably the most important structure in the cell because without the plasma membrane, the whole cell just falls apart. The plasma membrane is a thin surface that surrounds the cell, as I've highlighted in the bottom left, and it separates the cell from its external environment. You can think of it as the skin of the cell if you like, or the soft outer wall according to Randall Munroe. In addition to forming a barrier, the plasma membrane is also selectively permeable because there are proteins embedded in the membrane that control what can go into and out of the cell. It also has receptor proteins that can sense the external environment and help mediate the cell's response to changing conditions. At the molecular level, the plasma membrane is made up of a lipid bilayer which forms a hydrophobic barrier, blocking charged and polar molecules from passing freely into and out of the cell. In order to understand the function and behavior of the plasma membrane, it's important to know that it behaves as a fluid mosaic, meaning that the individual lipid molecules and embedded proteins are free to diffuse in the two-dimensional plane of the lipid bilayer. We'll talk a lot more about the properties of the plasma membrane and other cell membranes in our lecture next week on lipids. The next organelle I want to talk about is the nucleus. This is the largest organelle inside the cell, with a typical diameter of around 5 microns. As you can see on the bottom left, the nucleus is usually located close to the center of the cell, but not always exactly right in the middle. The function of the nucleus is to separate our DNA from the rest of the cell. Everything outside the nucleus but inside the cell is referred to as the cytoplasm. The cytoplasm will include all the other organelles that I'll discuss later in this lecture, suspended in an aqueous solution of soluble proteins and other biomolecules. This aqueous solution is called the cytosol. You can think of that as being short for soluble cytoplasm. The nucleus physically separates the process of transcription from the process of translation. Transcription happens in the nucleus, and translation happens in the cytoplasm. Now, I'm going to go over a few key parts of the nucleus, starting with the nucleolus, which is inside this yellow oval on the right. This electron-dense region is the site of ribosome biosynthesis. We'll go over ribosomes and the job they perform in the cell later in this lecture. The second part of the nucleus I want to highlight is the nuclear envelope. This is what separates the nucleus from the rest of the cell, and unlike many other membrane-bound organelles that we'll discuss today, the nuclear envelope is made up of 2 layers of lipid bilayer membranes, not just a single lipid bilayer. You can see these 2 layers of membrane in the yellow rectangle on the transmission electron micrograph on the right. The last part of the nucleus I want to talk about are nuclear pores, which are holes in the nuclear envelope. These are highlighted by the arrows on the electron micrograph. Nuclear pores, which we'll cover in more detail later in the semester, are large macromolecular structures that allow protein, RNA, and other macromolecules to enter and exit the nucleus. And as I mentioned before, the diameter of a typical mammalian nucleus is about half the diameter of the entire cell, on the order of around 5 microns. Another organelle is the smooth endoplasmic reticulum, or smooth ER. Let's zoom in here for a second. One of the key features of the smooth ER is that it's an interconnected network of membrane tubules and vesicles, as nicely illustrated in this picture here. This organelle plays many roles inside the cell. It's a location for lipid synthesis, and it's also a site for calcium ion storage. This becomes important for cell signaling, as we'll discuss later in the semester. We generally refer to the space inside the ER membrane as the lumen. The ER lumen also contains several components that are important for detoxification. It's the site for drug metabolism and other processing in the cell. And finally, the smooth ER plays an important role in the cell secretory pathway, which we'll talk about later in this lecture, and then go into greater detail later in the semester. To continue our discussion of organelle scale, the diameter of each tube and vesicle in the smooth ER is approximately 100 nanometers. The rough ER and the smooth ER are really just subcompartments of the same organelle, the endoplasmic reticulum, but it's useful for us to think about them separately. What distinguishes the rough ER is that its membranes are studded with ribosomes, which give it this characteristic rough appearance as seen in this electron micrograph on the right. These ribosomes synthesize protein, and this is why the rough ER is the site for membrane and extracellular protein synthesis. They're synthesized here and then transported throughout the cell by a vesicle trafficking system that we'll talk about later in this lecture. As I said earlier, the rough ER and smooth ER are contiguous, part of the same larger endoplasmic reticulum organelle. Unlike the smooth ER, which is primarily tubules, the rough ER is composed of flattened stacks, which are also approximately 100 nanometers in diameter. Okay, so we were just talking about ribosomes and the rough ER, and now I want to go into more detail about ribosomes here. But first, let's go back to the central dogma just for a second. DNA is transcribed to RNA in the nucleus, and the messenger RNA molecules have to move out of the nucleus into the cytoplasm before they can be translated into protein. The machinery in the cytoplasm that translates messenger RNA into protein is called the ribosome. Ribosomes are not technically organelles because they're not membrane enclosed, but they're similar to organelles in a lot of ways because they're large, insoluble macromolecular complexes that perform a specialized role inside the cell. And that role is to synthesize protein using messenger RNA as a template. Ribosomes themselves are structurally composed of a different type of RNA called ribosomal RNA, as well as many different protein subunits. All of this comes together. The ribosomal RNA and the protein subunits come together to form a ribonucleoprotein complex that's pictured in the top right So that's the entire ribosome bound to an mRNA molecule in the act of protein synthesis. Ribosomes are made up of a large and small subunit, which are labeled 50S and 30S in the top right figure. The 50S is brown and the 30S is blue. Don't worry about that now, though. We'll go over more details about this in our lecture on translation later in the semester. Okay, but one thing I do want you to be aware of is, depending on which mRNA they bind to, ribosomes will either be attached to the rough ER, where they synthesize membrane-bound and secreted proteins, or they'll be free in the cytosol, where they synthesize cytoplasmic proteins. The size of the ribosome is quite small in comparison to other organelles we discuss in this class, at approximately 10 nanometers in diameter. After proteins are synthesized in the rough ER, they're trafficked to the Golgi complex by vesicle transport and fusion. The Golgi complex is where all sorts of covalent modifications occur, as well as sorting and targeting to the proper final destination. The Golgi complex, also referred to as the Golgi apparatus, can be visualized as a series of flattened stacks, and there are different modifying enzymes at each level. Proteins synthesized in the rough ER are transported through the Golgi complex from the inner side to the outer side. We refer to these as the cis and trans sides. Cis is closest to the nucleus, and trans is closest to the plasma membrane. And as they travel through these stacks from cis to trans, they're modified in different ways at each level. Ultimately, they butt off into vesicles that facilitate their transport and targeting, either to the plasma membrane for secretion or to other membrane-bound organelles within the cell. As you can see, the size of each Golgi complex stack is on the order of the size of the rough ER and smooth ER at approximately 100 nanometers. Alright, so now we can put together all the organelles we've gone over so far to follow one specific targeting pathway, the secretory pathway. To go from start to finish, genes are transcribed into mRNAs in the nucleus, and mRNAs are transported through nucleopores and then localized to ribosomes on the rough ER. There, the mRNAs are translated into proteins, and these proteins are then trafficked by vesicles to the Golgi apparatus, where they are covalently modified in different stages before further trafficking, and finally secretion by vesicle fusion with the plasma membrane. This vesicle fusion is called exocytosis. In addition to protein trafficking out of the cell by exocytosis, there is a reciprocal process called endocytosis that internalizes material from the extracellular environment. This is illustrated in the electron microscopy images on the right. One more thing that I'd like to note on this slide is that the rough ER is contiguous with the nuclear envelope. They share the same membrane. This illustrates another thing that we can see on this slide, and that's the topology of membranes in the cell. This is an important concept, so let me explain what I mean here. Outside of the cell is an oxidizing environment, but the cytoplasm is reducing. This has important functional consequences for proteins that we'll talk about on Thursday. And so what I mean by, say, membrane topology is that, as you can see in this picture, the lumen of the secretory vesicles, the Golgi, the ER, and the nuclear envelope, all of these are linked to each other. They're either physically contiguous or linked by vesicle transport and fusion events. And therefore, what that means is the lumen of all of these structures is oxidizing, just like the extracellular space, as opposed to the reducing environment of the cytosol. And this will have important functional consequences for protein structure that we'll talk about more on Thursday. After endocytosis, many of the internalized materials are destined for degradation, and endocytosed vesicles are trafficked/converted into a specific organelle that specializes in degradation, called the lysosome. This organelle is responsible for degrading material brought in by endocytosis, as well as other cell components that are targeted to the lysosome from within the cell. One key feature of lysosomes is their acidity, with a low pH of approximately 5. Lysosomes also contain specialized digestive proteins called acid hydrolases that catalyze a hydrolysis reaction. Hydrolysis is a water-mediated cleavage that can break down the covalent bonds of proteins and other biomolecules. This is the exact reverse of the condensation reaction that we talked about earlier in the polymerization of nucleotides, saccharides, and amino acids. Acid hydrolase catalyzed breakdown and degradation can also happen to entire organelles. If they're damaged and no longer functioning, they can fuse with the lysosome and then be degraded, as shown in this electron micrograph. Returning to our order of magnitude analysis, lysosomes can vary a lot in size, but they're typically on the order of hundreds of nanometers in diameter, although they can go up to 1 micron or more. Another important organelle in the cell is the mitochondria. This is the powerhouse of the cell, where ATP is produced. We'll talk about this later in the semester in our lectures on metabolism. Mitochondria primarily use glucose metabolites as fuel for ATP production, but they can also use lipids as well. Key features of the mitochondria include a double membrane similar to the nuclear envelope. Here we have an outer membrane and an inner membrane that includes invaginations called cristae. Everything in between the cristae is called the mitochondrial matrix. As a side note, there's very strong circumstantial evidence that mitochondria were once freestanding prokaryotic organisms that were endocytosed by a eukaryotic cell and then developed a symbiotic relationship rather than being degraded. This was a really good bargain for both cells. The eukaryote gained a dedicated power supply, and the prokaryote gained a safe environment and steady supply of nutrients. The strongest evidence for this evolutionarily ancient symbiosis event is that mitochondria have their own DNA and ribosomes, which are closely related to some present-day prokaryotes. I want to point out that in live cells, mitochondria exist as a dynamic set of interconnected tubules that look kind of like this. This is more representative of what they look like, more like a lava lamp rather than the static jelly beans that are often depicted in cell biology textbooks. And the diameter of a typical mitochondria is on the order of a few microns. All right, so we've covered a lot of the important organelles in the cell, but what about everything else? Everything else is the cytosol. This is the soluble portion of the cell that's enclosed by the plasma membrane, but not part of any organelle. The cytosol is an aqueous solution of proteins and other biomolecules, and it doesn't include organelles or any other suspended particles, such as ribosomes or another component that we haven't talked about yet, cytoskeletal filaments. The cytoplasm, on the other hand, is everything inside the cell except for the nucleus. So the cytoplasm includes the cytosol and the organelles and all of the insoluble particles like ribosomes and cytoskeletal filaments. We'll cover the cytoskeleton later in the semester, but to come back to our discussion of scale for different components of the cell, cytoskeletal filaments are on the low side, smaller than even ribosomes, as illustrated in this electron micrograph on the right. So cytoskeletal filaments, there's different types of these, but they're on the order of a few nanometers to tens of nanometers in diameter. Okay, so to conclude this section on organelles and structures in the cell, I want to summarize our order of magnitude analysis because it's important to get an idea of how these organelles scale to one another, as well as how they scale to an absolute perspective. So a mammalian cell is on the order of 10 microns, and the nucleus is about half that, on the order of 5 to 6 microns. After the nucleus, the next largest organelles are the mitochondria and lysosomes. These are on the order of approximately 1 micron each. Next come the ER and Golgi organelles, which are an order of magnitude smaller, with their individual stacks and vesicles on the order of 100 nanometers in diameter. After we go down another tenfold, we get to the smaller insoluble particles. These are not strictly speaking organelles, but are instead macromolecular complexes like ribosomes and cytoskeletal filaments, which have a diameter of approximately 10 nanometers. To put these scales in the context of the molecular building blocks that we talked about earlier in the lecture, a typical protein has a diameter of a few nanometers, and a single carbon-carbon bond has a length of around 0.1 nanometers. All right, so that's the end of our section on organelles. We're going to wrap up the lecture now talking more about viruses and the coronavirus pandemic. The virus that causes COVID-19 is SARS-CoV-2, which is a coronavirus. And you can see in this diagram how those compare to other types of viruses. Coronaviruses have RNA genomes instead of DNA, and that RNA genome is single-stranded rather than double-stranded. Viruses travel from host to host as viral particles called virions, and the coronavirus virion particle is enveloped rather than naked, meaning it's enclosed by a lipid bilayer. Here's a zoomed-in view of the SARS-CoV-2 viral particle, with its RNA genome surrounded by a lipid bilayer containing various proteins. On the right, this diagram shows its viral life cycle, starting by protein-protein interaction on the surface of the plasma membrane that facilitates internalization by the process called endocytosis, which we discussed earlier. After endocytosis, the viral membrane fuses with the membrane of the internalized vesicle, allowing the viral RNA genome to escape into the cytoplasm. And based on our earlier discussion of the central dogma, it shouldn't be surprising what happens next. There are a few intermediate steps where the genomic RNA is processed, and then the viral RNAs are translated into viral proteins that assemble into new viral particles, and each viral particle packages up a new copy of the viral genome before it's secreted to go on and infect other cells. The viral life cycle for SARS-CoV-2 is pretty typical for a virus, and we can generalize the life cycle of viruses here. Number 1, enter the host cell. Number 2, hijack the host cell machinery to replicate. And then number 3, release viral particles to infect more cells. A question that I think is interesting is, are viruses alive? To answer this question, we have to think carefully about what our definition is for life. What does it mean to be alive? What are the criteria? We talk about the viral life cycle, but this is very different from other organisms that we consider to be alive. So I ask you this, what's your definition of life? And do you think viruses fit into it? I'd recommend taking at least a few minutes thinking about your own definition that you can come up with before looking for answers on the internet or in a textbook. Maybe some of you have seen this question before and can try to jog your own memory before looking it up. Once you come up with your own answer, you might find these videos interesting. They won't give you a complete answer to the questions above, but they do provide some interesting background information. All right, that's the end of the lecture for our first class. We covered a molecular parts list for the cell, the central dogma, and the major organelles and structures inside the cell. I also gave you a very brief introduction to the COVID-19 pandemic and an overview of the SARS-CoV-2 viral life cycle. I'm looking forward to hearing your thoughts on whether you think viruses are alive and how you define life in the first place. Thanks for watching, everyone, and I'll see you in class on Tuesday.